Hello everyone, how are we doing today? So in today's video, we're going to be going over the major macromolecules of life. So we talked about these a little bit in the lecture, but just as a reminder, remember there are 4 major macromolecules in life. And the goal of this little lab video here is to make sure that if you look at an image of one of these molecules, you can identify what group it comes from. So there are 4 groups. First ones are the carbohydrates. These are our sugars. So we're going to look at those. These come in monosaccharides. So monosaccharides. They could be disaccharides, or they could be polysaccharides. So di is 2, mono is 1, poly is many. And these are all different building blocks of sugars. Second group we're going to talk about today are the lipids. These are our fats. So fats. We're going to talk about 2 major groups today. We have our triglycerides, and then we have our phospholipids, which make up our cell membranes. There are also steroids, which are all cholesterol-based, and also eicosanoids. But we're just going to draw triglycerides and phospholipids today, because mainly understanding those, you can understand the rest as well. And then we have proteins. Proteins are our enzymes, our structural proteins, hormones, some hormones, not all hormones, but proteins serve many of the functions in our body and make up what we are. And then to decide what we are, we have our last macromolecule, which are our nucleic acids. Remember, nucleic acids can be DNA or RNA. So ribonucleic acid or deoxyribose nucleic acid, they differ in the carbohydrate that makes them up. So nucleic acids have a carbohydrate subunit in them. So these are the major macromolecules. So the goal of this little laboratory video here is to be able to identify them, because then that helps you understand what they're going to do in the body and how they're going to work in the body. So let's go to the first one here. So these are the carbohydrates. Remember I said the carbohydrates could be monosaccharides, disaccharides, or polysaccharides. So just a simple example here. So carbohydrates are always a ring structure. Now, when you draw them on paper, they could be what are called linear or bridge structure, but don't worry about that in this class. So here is the general example of a carbohydrate. So each one of these is a carbon at the corners here. I know it's not a great representation, but we're drawing glucose here. So glucose has a hydrogen, hydroxyl. Now, again, you don't have to remember all these parts. I'm just drawing them to show what it looks like. And then up here, we have an HOCH2 group. So it connects to that carbon right there. So this is glucose. This is a monosaccharide. So carbohydrates have this notorious, so the glucose is C6H12O6. So 1, 2, 3, 4. Yes, C6H12O6. Oh, there's the 6. I was like, where's the 6 hydroxyl? I forgot it right there. I'm like, I know it's 6, and I was only counting 5. I was missing an oxygen right there on that one. There's also a hydrogen that comes off this one here that I forgot to draw. Okay, so there was the one I was missing. I'm not freaking out here. Glucose is C6H12O6. They all carbohydrates all have this similar makeup. So C, N, H2N, O-N would be a brief way you could write that. So here, this is a monosaccharide. Now, if you connect 2 of these together, so 2 glucoses, now I'm not going to draw all the hydroxyls and so forth. I'm just going to draw a generic structure here. So just that, they can connect to another glucose, and then it forms a disaccharide. So now I'm going to draw another one right here. Again, not perfect, but general representation. This bond that forms between these is called a glycogen bond or a glycosidic bond. And this is called maltose, example of a disaccharide. Maltose, not tase. So maltose is this disaccharide. Now, if you combine a whole bunch of these together, You then form, so now I'm just drawing O's to represent the glucose. So now all these little glucose chains chain together, forming a highly branched, so these can become branched, come down, and it's a high storage structure capabilities within this branched glucose structure. This is called a polysaccharide, or better known as, in our body, glycogen. Plants can also do this as well, and in them it's called starch, or amylose. Amylose. Cellulose is another example of a bunch of glycogens chained together, except their bonds are different, so we can't digest them. We can digest starch and glycogen, but not cellulose. We can't absorb that. Then one carbohydrate that you might know of that causes problems, lactose. Lactose is a disaccharide, and the problem with lactose, so lactose is a disaccharide with the monosaccharides, whether they galactose and glucose, and requires an enzyme called lactase to break this glycosidic bond. Individuals who are lactose intolerant don't have a functioning lactase, so this lactose goes through undigested and causes a couple different issues. So these are a general representation of the carbohydrates. Next, I want to go into the lipids. So here, lipids are our fats. So we have two main types I want to draw here. First one is a triglyceride. So these are the main energy storages of the cell. So a triglyceride is made up of a glycerol and 3 fatty acids. So here, let's first draw a glycerol. A glycerol has 3 carbons coming down, a 3-carbon chain, and then right here, there's a hydroxyl group attached to each carbon, and then there are hydrogens surrounding the remaining free bonds. Now, each of these hydroxyls right here can form a bond with a fatty acid. So a fatty acid has a carboxylic acid end. So then here would be an OH group, double bond oxygen. That's a carboxylic acid right there. And then a hydrocarbon chain. I'm just going to draw these as CH2s. Now these could have some double bonds in them as well, but I'm just going to represent them like this. And that could be indefinite, so you can put a bunch of periods there. So there'd be 3 of these down through here. I'm not going to draw all 3 to save time here. And then where does this reaction take place? So this forms what's called an ester bond. It attacks this hydrogen within this hydroxyl group and this hydroxyl group right there. This reaction, when this happens, removes water because it's a dehydration reaction. And then I'm just going to draw the top one here. You'd have a hydrogen, hydrogen, and then right here would be the ester bond. So it goes from the oxygen right to the carbon. That carbon then has this double bond oxygen sticking off of it, and then you have the hydrocarbon chain. And then you'd have that here as well. There's the next one, hydrocarbon chain. And then here would be the next one, bond, double bond O, hydrocarbon chain. And then, of course, there'd be hydrogen. So this is the general representation of a triglyceride. Main energy storage things here. So when you're looking for a triglyceride, look for a long chain of hydrocarbons. Now, I kept this spot empty down here for another reason, because I want to erase this part here and just leave the middle one. Because now this is where we get the next one I want to talk about, which is a phospholipid. A phospholipid is what makes up our cell membranes. So here we typically draw these as these little circles and with these tails, and this would be the polar end, and this would be the nonpolar end, and that's what forms our cell membranes. And again, it's the polar-nonpolar interactions and being in an aqueous environment of water that allows this to happen. So how does this bond form? So here, another fatty acid would come in. Here's a carbon, double bond oxygen, and let me erase that little arrow right there. And then this would be the hydrocarbon chain, ... Now down here, something different happens. What comes in is called a phosphorus-containing group. I'm not going to draw this whole group, but it's where it gets its polarity. So it has a phosphate on it. A phosphate is P, double bond oxygen. There's an oxygen here, has a negative on it. Actually, this one might be a hydroxyl. This one then would have a negative sign on it, oxygen, and then for this one has a CH2, so slightly hydrocarbon part here, but not much. So 2 carbons, and now it has a choline head. What a choline head is an N plus, and then it has 3 methyl groups. CH3, CH3, and then CH3. So what this is, it's very highly charged. Here we have negatives on the oxygen, and here we have an electronegative nitrogen as well. So this forms a bond with this as well, same as these ones up here. However, this one now turns away because of these charges from these tails. So this, I can't fit it in here. I'm just going to draw this head coming off as a P, forms that phosphate head. So in reality, how this structure looks, this is that phosphatidylcholine head up here. This is where the phospho part of it comes from. Then this is the lipid part in the tails here. So same bond, very similar structure to glycerides, except just slightly different on this last carbon on this glycerol, where this phosphate group or phosphatidylcholine group gets added right here. So kind of neat how that works. That makes this side polar and this side non-polar. Now, just understand that P would be here, so this whole molecule would be flipped. I just ran out of some space here, so we work with what we have. All right, that's that group. Next group are our proteins. These ones are my favorite. They're most diverse. You can have globular proteins. You can have structural proteins. They can be enzymes. You know, the keratin in our nails, the keratin in our hair, collagen. All of these are albumin transporting things in our blood. So many different kinds of proteins. We're going to talk about so many proteins this semester, so it's important to understand. During the lecture, we talked about the 3 different structures of proteins. They come in the primary structure, which is just a chain of amino acids. Remember, proteins are made up of amino acids. So an amino acid has a central carbon. It's called the alpha carbon. And then it has a carboxylic acid end. Then there's a hydrogen that comes off the bottom here on most. And then over here is the amine group, which is an NH2 group. So this is the general structure of an amino acid. Right here is what's called the R group. Now, there are 20 different amino acids in our body, but you want to look for this alpha carbon, this carboxylic acid, and this N terminal. So this is called the C terminal, this is called the N terminal. Every amino acid has this, every polypeptide chain. So what happens, let's say this is now represented as a little circle. You form a whole bunch of these amino acids in a chain. Now that becomes a peptide, and that can fold in from this. So this would be the primary structure, and then it can form alpha beta-pleated sheets and alpha helix structures, which are a secondary structure. And then these R groups can interact. These R groups can be charged. They could be a positive charge, a negative charge. They could be polar, non-polar. They could be acidic. They could be basic. And those then, or they can form covalent bonds with each other. So that then forms the tertiary structure. Now, some proteins require multiple subunits to come together, so you get multiple tertiary structures coming together to then form a functional quaternary structure in the protein. But here, let's just diagram out a simple peptide bond to show what one looks like. So here you have the central carbon. So this is just 2 generic, let's make it a glycine. There'd be a hydrogen there, the easiest one you could choose if you wanted to make one here. So let's make that a glycine here too, which is just a hydrogen. So now we want to form a peptide bond. Same type of bond here. So we have a reaction occurring between this hydrogen and this hydroxyl group. So this reaction comes down. Guess what's removed? It's a dehydration reaction. So water is removed. That carbon now goes to that nitrogen. That's where the bond forms. We have a double bond connected to that, and then we have the remainder here. So there would be the glycine on this side. There's the C terminal, or carboxyl terminal, and then we have the remainder on this side. There's the hydrogens, and then here's the N terminal. So right there, this bond is then the peptide bond. This is then called a dipeptide. Kind of cool how proteins work. So all your proteins in our body are thousands of amino acids long, so thousands of peptide bonds between them. You can break these then via a hydrolysis reaction, which uses this water and brings it back in, breaks that bond, and you can separate them into different amino acids. So when you eat proteins, your digestive system, you have enzymes that are called trypsinase, and there's carboxypeptidase, there's different peptidases. Bromelain, which is found in pineapples, actually can break these, is an enzyme that can break these bonds too. It's a denaturant. So it can break these bonds, and you have to break them down to the individual amino acids to be able to absorb them in your small intestine, and then they go to your body and then build up into other proteins. So very, very important. Talk about proteins a lot, especially in the muscles. Actin and myosin will be two of your favorite proteins once we get there. Okay, now finally we get to the last group, which are our nucleic acids. So remember, nucleic acids are the DNA and the RNA. Now, so they're both made up of a sugar, which is either a deoxyribose or a ribose. They have a phosphate and a base. So a sugar and a base is called a nucleoside. A sugar, phosphate, and a base is termed a nucleotide. So first, let's draw the sugar. So here, it's a ribose. So it's 5 carbons. Boom. And then up here this is where the bond comes off. Well, now I want to finish this because here I have 2 hydroxyl groups. 2 hydroxyl groups right here makes this a ribose. If this was an oxygen, just an oxygen right here on this one, it'd be a deoxyribose. Right here's a carbohydrate if I would stop right there. However, a bond forms here, and this one's a little bit more difficult to draw. And then like this is a double bond. This one's a double bond here too. And now we form that one. This is a nitrogen right there, and nitrogen right there. I know it's not pretty, but I just want to get the job done. So this is actually an adenine base. So here we have an adenine base. There's a hydrogen there too. So ribose, adenine. Again, so this is also known as adenosine, but that's a nucleoside. So each base, A, T, C, and G, would come in that form for both DNA and RNA. Well, not T in RNA. It'd be a uracil in RNA. So we have that difference too. Now, the last thing we need to draw here is the phosphate, which comes off right here. So right here, we have this. Now, I never drew that last carbon. I was waiting to draw it. So we have that last carbon coming off here. Remember, this was a CH2OH. That OH forms a bond to a phosphate group then. So here, oops. Thought I hit the eraser. So we form a bond to this phosphate group. I should have just drawn a circle with a P. Would have made this much easier. And then there's an O negative, and then here's an OH. Now, right there's the phosphate. So there is your typical nucleotide. So that's the general structure you want to look for. Now, the base will change. It can be, like I said, it could be an adenine, could be a thymine, cytosine, guanine, or uracil. And the ribose could be a deoxyribose if DNA as well. Now, there's one last thing I want to talk about here was what if, and this is the reason why I drew RNA, what if we had another phosphate and another phosphate? You know what this structure is? This is ATP, or adenosine triphosphate. You know, that's our energy currency. You remove one, you have adenosine diphosphate. You remove another, you have adenosine monophosphate. So a nucleotide of RNA with adenine is very similar to AMP, adenosine monophosphate. So not a lot of people realize how similar ATP is to an A base in RNA. You know, very similar structure, and they kind of go hand in hand with each other. But those are the major macromolecules. I know there's a lot of structures, but like I said, you want to be able to look at a structure and be like, oh, that's a carbohydrate, that's a lipid. I know I didn't draw cholesterol or eicosanoids, but they are still lipids that are important as well. Or you're looking at a protein, be like, oh yeah, that's a protein. I can see the different structures in that protein. It looks like a fibrous protein compared to a globular protein. We'll talk about that more in the class as well. And then lastly, nucleotides here to be like, oh yeah, that's DNA or RNA. But that's all I have for today. If you have any questions, feel free to reach out to me. But I hope you all have a great day, and bye-bye.